have learned about the protein. You know that they are made up of amino acid. You know that they are present and they are active when there is a three-dimensional form. So all these characteristics which you know about protein, that you will see is playing enormous role in the activity of enzymes. Now, why I said nearly enzymes? Because there are enzymes, they may not be proteins in nature, that they may be some other biological molecule. So I'll give you an example here. Earlier, when we studied around that time, there was a notion which was said that all enzymes are proteins. Then it was not nearly all. So now there are few enzymes which are made up of RNA. So they are known as ribozymes. So ribozymes are basically the RNA molecule which folds and they give the activity of enzyme. So this is the some of the exception, but otherwise all the enzymes, they are proteins in nature. Now tell me, what could be the difference between enzymes and proteins? Of you see, the difference between proteins and enzymes are by nature, by their structure, both are same. The only difference is that the enzyme, they have a site which can catalyze the reaction, and that site is known as an active site. So the proteins with catalytic power are enzymes. Is it clear to you?
Yes, sir.
Okay. So protein as a class of macromolecule are highly effective catalysts for enormous diversity of chemical reaction. So there are many type of reaction. I'll show you that at least 6 different classes of enzymes they are present. They show different types of chemical reaction. So if you look at the reaction catalyzed by the enzyme, they will be like any chemical reaction. So they are helping in attainment of the equilibrium. And why it is so? Because the proteins, they have a capacity to specifically bind a very wide range of molecule, and because of this binding affinity of the proteins with the different molecules, they have catalytic power. And they also catalyze reaction by stabilizing transition states. I'll talk about this concept, what is the stabilization of transition state, and the high energy species in the reaction pathway. So what they do is that actually this part you have learned in case of catalyst as well, that there is a transition state analog which is formed, and that is what is being stabilized, and that's why the activation energy is lower. Now typically how enzyme looks like. So I am showing you an enzyme which is a very common enzyme which is present in our stomach, which is our gastric juice, which is chymotrypsin. The name of enzyme is chymotrypsin. Its job is to break down the protein which we digest. And look at this. I'm showing this as one of the model
So,
which is
are you
showing.
sharing the screen because it's not visible?
It may take a second because this slide is quite heavy.
Okay.
Just wait for a few seconds. If it is not visible, maybe I will come to this. Now, is it visible now? I have come in a different mode, normal viewing
Yes.
mode. Let me know once it is visible. Otherwise, I'll move on to another slide. It's a heavy slide. It shows three-dimensional structure of a protein. And in the protein, I have shown the active site where the substrate is binding. So if not visible, I'll move on to another slide because it's a heavy slide. It may be disturbed because of the network connectivity. Okay, I'm moving to the next one. Is it visible?
Yes, sir, we can't see it.
No, sir.
Oh, I will connect with another network in that case, and it will take few seconds, and then probably we can you can see it also. Is the screen visible now?
Yes, sir.
Okay, just a second. I'm just looking at, can we show the enzyme slide? Okay, tell me if you can see the slide.
Yes, sir, we can.
Okay, because of the heavy picture, probably the transmission was not possible. Now, looking at this enzyme, you see that it is nothing but three-dimensional structure of protein. Number one. Number two is that there are very few residue which are involved in the binding. Rest of the molecule is, free. So this enzyme, just this portion, which is red color, they are the key active site. So I'll tell you what are the active site. Just go back to this. Yeah. And then it shows the substrate, which is binding. Now, don't get confused. In case of chemistry, reactant here we call as substrate. So here we will call substrate and product, there you call reactants. So what you see is that there is a whole structure of enzyme which is here, which may be having say 1,000 amino acid involved. But the one which is involved in the active side, they have very few amino acid, maybe 5, maybe 6. So I'll explain this much better in the coming slides. So next one shows that the enzymes are powerful and highly specific catalysts. How they are highly specific, that part we'll discuss. The enzymes are highly specific both in the reaction that they catalyze and their choice of reactant, which are called substrates. So why I'm saying so is that one thing could be that you have an enzyme which catalyzes the bond between COH and NH2. So here, where do you have this kind of a bond? There is a peptide bond. Where do you find peptide bonds? These are present in
Mhm.
proteins. So if I have to catalyze a reaction, so one specificity could be that it catalyzes a peptide bond cleavage. So suppose I have to hydrolyze a protein, what I require is I require something which can break the bond between carboxylic group and amino group. So here we are talking about the enzyme which catalyzes the cleavage of COH and NH2 group. That is a peptide bond. Now, then it says that the specificity of an enzyme is due to the precise interaction of the substrate with the enzyme, and this precision is the result of intricate 3D structure of enzyme and protein. So what you have here is that the the choice of reactant, that means a reactant or a substrate should have CONH bond, that means peptide bond, then only the enzyme will catalyze. It will not catalyze a reaction. For example, it will not break the phosphodiester bond. Where do you find phosphodiester bond? In nucleic acids. It will not catalyze other type of ester bond. So what it catalyzes, very specific bond when you have COH group and NH group as the product. Now, another thing which you see in this on the right hand side is that it is a catalyzing when the carboxylic group is either lysine or arginine. Both of them are basic amino acids. So if this group, which is in the carboxy terminal, is a lysine or arginine, then only your enzyme will catalyze the reaction. So chymotrypsin, they will catalyze the reaction. Trypsin will catalyze the reaction only when your lysine or arginine is present at the C terminal. Now your question may be, suppose lysine is present, suppose this one where the bond is broken, if it is present instead of this one. So if it is present on this amino acid which is a lysine, reaction will not be catalyzed. That means it is very specific when it is present in the C terminal. You understand what is the C terminal and N terminal of amino acid. Now, another specificity you see that here it is another enzyme which cleaves the bond only when arginine is present. It will not catalyze a reaction when lysine is present. So, you know, the specificity is a very, very highly specific nature of the enzyme. So, specificity is very, very important here, which you will not find in chemical catalyst. So, can you tell me why they become so specific? What could be the reason? You have seen the previous slide where you have seen that amino acid, they fold, they give you three-dimensional structure. In three-dimensional structure, you have created a site which is coming from different amino acid, coming from different parts of the protein, and then it binds. So the specificity comes because of the three-dimensional nature of enzyme. That is one point. What else? Why should they become stereospecific? Can anyone tell? Now I have told you that they are specific. They are specific. Why they are specific? Because they can bind in a very specific mode in three-dimensional structure. Now question comes that why they become, stereospecific. When you look at the enzyme structure, they are made up of what? They are made up of amino acid. And what are the amino acid? Amino acid is a group of different atoms where the stereospecificity exists. Except for glycine, all the amino acid, they are stereospecific in nature. And being stereospecific, that means in three-dimensional, they have stereospecificity in terms of binding with the substrate. And that is why you find the stereospecificity which does exist in enzyme. So if enzyme catalyzes a reaction where R form is present as a substrate, it will not catalyze a reaction where S form is present. So it is very specific for one stereospecific molecule. So as I said, the top one, number A, is showing you trypsin, which is a digestive enzyme, and B is showing about the thrombin. It is an enzyme which is participating in blood clotting. So these two gives you an example of the specificity, and that means it is not just specific for CONH bond, it is also specific what is contributing towards the CO. That means the amino acid which is having this COH group. Now, so far we have talked about the protein part of the enzyme which acts as a catalyst. Now, other than the protein, there are certain enzymes which may involve a cofactor for their activity. So now you may have a question that do all the enzymes require this cofactor for their activity, or there are very few enzymes which require the cofactor for their activity? So for example, in the previous slide, when I talked about trypsin, this one, which is catalyzing the bond cleavage of this, or chymotrypsin discussed earlier, this one, all of them, these proteases, they do not require any cofactor. So there are a set of enzyme which does not require any cofactor. Their protein part is capable of catalyzing the reaction. So the catalytic enzyme of many catalytic activity of many enzymes depend on the presence of a small molecule termed as a cofactor, although the precise role varies with the cofactor and the enzyme. That means how much and what kind of enzyme is playing a role, that means protein part is playing a role, and how much cofactor part is playing a role. So what it means when I say that in certain cases, both protein part as well as a cofactor is required for the enzymatic activity. It means that if just protein part is present, it will not work. If just cofactor part is present, they will not work. So for the functionality, you require the cofactor as well as a protein part. So in such enzymes, the protein part is called as apoenzyme. The cofactor could be organic molecule or a metal, and both of them, when they become functional, they are called as a holoenzyme. Now, coming to the cofactor, as I said, they could be metal or they could be organic molecule. These metal, I will talk about. What are the metal? Can you tell me any enzyme where metal is involved? We have discussed it in the previous classes. You have learnt about the sodium potassium ATPase. This is an enzyme. Of course, it is acting as a transporter, but they require sodium ion or potassium ion for their activity. If they are not present, they will not work. So they are the enzyme requiring cofactor. So what is the protein? Protein is the ATPase part, and the cofactor could be sodium ion or potassium ion. So these cofactors, when they are present as an organic molecule, they are called as a coenzyme. Otherwise, they are metal ion. Now, sometimes these coenzymes, in fact, when I talked about the coenzyme, this coenzyme is always part of the cofactor. So we can have a plus coenzyme acting as a holoenzyme. What does it mean? It means the cofactor is a general name which could be coenzyme. We call coenzyme when this cofactor is a organic molecule. Am I clear? Is it clear to you?
Yes, sir.
So coming to the another very interesting point, which is just for nomenclature, sometimes this coenzyme is tightly bound to the enzyme. That means we have to break a covalent bond to remove the coenzyme. And those times, these coenzymes, they're referred as a prosthetic group. So it is just tightly binding with the enzyme. That is what gives them additional name. So I have given this name just so that you do not, get confused with prosthetic group. So here, cofactor could be metal ion, it could be coenzyme, it could be prosthetic group. All these things, they constitute cofactor. Now coming to these cofactor, whether it's a metal ion or it is your coenzyme, all of them, they bind with the apozyme very loosely. That means they'll bind like a substrate and they can be removed. So as substrate and product, they are removed from the surface of enzyme, the cofactor can also be removed. But for the prosthetic group will remain joined with the apoenzyme. Another interesting point, what you find is that this coenzyme, all of them, they are coming from a source, and this source is known as a vitamin. So, and that is why at the end of the chapter, we'll talk about the vitamins as well. Though today, I'll give you some listing of the vitamins, but in general, we'll talk about the vitamins in the next few classes. So this is the list which provides you the name of coenzyme, for example, thymine pyrophosphate, flavine adenine dinucleotide, NAD, pyridoxal phosphate, coenzyme A, and so on and so forth. For example, here I have given you the chemical name. On the right hand side, it is the name of enzyme. So you can see the coenzymes are also specific with the enzyme. It is not that thymine will work as a coenzyme for all other enzymes. So their specificity is linked with the specific class of the enzyme where they catalyze a reaction. More of this I will talk about later. But the question comes from where this coenzyme is coming. These coenzymes, they are coming from the vitamin. So for example, vitamin B1, so they can be called as a thymine, and thymine is modified to coenzyme, and then it acts with the enzyme. Now there is a list of metal ions which act with the enzyme. So for example, zinc with carbonic anhydrase, a carboxypeptidase, magnesium with some of the enzyme, hexokinase with magnesium, and so on and so forth. So you have several metal ions, they act as a cofactor during enzymatic reaction. They are bound on the enzyme surface at the active site, and that's the way they catalyze the reaction. This part I have already discussed, that enzyme may transform energy from one form to another. So there are many examples which has been given. It is not necessary we discuss about them. We'll discuss about enzymatic formation of energy from one form to another form. But it is very clear that they can convert the chemical energy into mechanical energy into the chemical energy, the transformation from one form to another form. Even in chemical energy, you can have a bond energy, you can have a highly specific, storage of energy in the form of bond. So we'll talk all about that. Now coming to the question that what are the types of reaction which can be catalyzed by the enzymes. Now there is a full detail about the enzymes, what kind of a reaction they can catalyze, but in general they can catalyze only 6 types of reactions, and they have been clubbed under different types of enzyme. For example, if any enzyme catalyzes oxidation reduction reaction, we club them under a class of enzyme which is called as oxidoreductases. So for example, lactate dehydrogenase. So if you see oxidation reduction reaction catalyzed by an enzyme, they will always come under oxidoreductase. Same way, if the transfer of the group occurs, they come under transferases. If hydrolytic reaction comes, they come under hydrolases. If the addition or removal of a group to double bond occurs, they are called as lyases. If isomerization reaction occurs, isomerases. And if there is a joining of the bond, ligation of the two substrate at expense of ATP hydrolysis, they are called as ligases. So all the enzymes, suppose I discover a new enzyme, and I have to classify, I have to put them under one of the class that will be dependent on the type of the reaction. So this classification has come from EC, which is known as Enzyme Commission. Like you have IUPAC in chemical nomenclature, here you have Enzyme Commission, which is a subset of IUPAC, which gives you all the name of the enzyme based on their type of reaction. I have not given a detail about the class and further subclass. So whenever you have oxidation reduction reaction, you may have a specific group which is involved in oxidation reduction reaction. You may have the specific substrate involved in the reaction. And last one is the exact reaction. So there will be 4 number. One will be for class. So any enzyme, I may not write the name of the reaction. I may not write the lactate dehydrogenase. I can give their number. So each enzyme can be given a specific number based on the enzyme commission, and that defines the class of the enzyme, subclass of enzyme, subclass of the enzyme, and type of enzyme. So you have a 4-digit number. One of the digit could be double digit also, but what I'm saying is that each will be classifying and giving the information about the enzyme. This is just for information, just for your knowledge. Suppose I give you a number 1.4.8.9, you should be able to decipher if I give you the list that what kind of reaction they are going to catalyze or what is the classification. Okay, is it clear to you? Have you understood the concept of this classification? It is based on the type of reaction they are catalyzing. Further, there will be subgroups in the types. For example, you may have a for example, if I'm talking about the transferases, number 2. So any transfer reaction will come under transferases. But suppose there is amino group transfer or carboxylic group transfer, that will give you subclass. Then I can have which substrate is transferring, that becomes subclass, and the final one will be based on the exact reaction, both substrate and the product. Anyway, that is not important. Just you need to know that yes, enzymes can be classified based on the reaction they catalyze. Now coming to some thermodynamics about the enzymes. There is a free energy which is useful thermodynamic function for understanding enzyme. So what are these free energy? How the free energy is involved in the enzymatic reaction? To understand fully about the enzyme, how they operate, we need to consider two thermodynamic property. One is a free energy difference, that is a delta G between the products and the reactants, and the energy required to initiate the conversion of reactant to the product. This part you have already done in chemistry. We will just focus on whatever is required for enzyme catalyzed reaction. So this is also clear that in any reaction, if delta G is negative, it is a spontaneous reaction, and this is a exergonic reaction. And if I say the delta G is 0, that means system is on equilibrium. And if I say delta G is a positive, that means you require free energy input for the reaction. And this can be calculated based on your previous understanding about the reaction. That means A and B, if I give you as a reactants, here we call them as a substrate, and C and D are products, or again we call them as a reactant. So this can be given by delta G, and delta G0 is a standard free energy chain where R is a gas constant, T is absolute temperature, and of course C, D, A, B all are reactant concentration. So any reaction will depend on the molar concentration of substrate and product. So suppose you are carrying out a reaction in lab in a test tube, the delta G value may be different. But if you carry out inside the body, the delta G value will be different. Why it is so? Can you tell me why it should be different? When I carry out a reaction, which you have seen in the biological reaction, for example, hydrolysis of, say, a protein, why should it be different in the lab and inside the body? The delta G value, why should they differ? They will differ because of one factor, that is the room temperature. So if room temperature is different than body temperature, it can affect the delta G value. What else can affect the delta G value? It could be the molar concentration of product and substrate. So substrate concentration inside the body will be different than you can take typically in a reaction in mixture in test tube. So there are always values which has been given for the biological system. So you may have a difference in the value which is calculated in a reactant present in the test tube and the one which is occurring inside the body. So that is what is given in the last point. The delta G of a reaction depends on the nature of reactant and their concentration. Now, this is the concept which we have talked a little earlier, is that enzyme cannot alter the law of thermodynamics and consequently cannot alter the equilibrium of a chemical reaction. What does it mean? It means that suppose I have interconversion of A into B. So if in the absence of an enzyme, so the first part is showing where the reaction is occurring in the absence of enzyme. If K1 or K forward is 10 to power the minus 4 per second and K reverse is 10 to power the 6 per second, what is more important here is what is the what is the equilibrium constant K. Now this part, if enzyme activates the reaction in the forward direction, the same rate by that or same fold, they will be increasing the rate in the reverse direction. So what will happen? If I increase the rate of reaction in the forward direction as well as in the backward direction, what does it mean? It means that equilibrium attains very fast. That is what we discussed. So it is the equilibrium constant which is achieving fast. Now the free energy difference between the reactants and products accounts for the equilibrium of the reaction. But enzyme, what it does is that it accelerates how quickly this equilibrium is attained. That is what we discussed in the previous slide. So a chemical reaction of a substrate S to a product P goes through a transition state. Question is being addressed that why it attains how it helps in the attainment of equilibrium, or in other words, how it helps in lowering of the activation energy. It is basically because if a substrate is converted into product, it is going through a transition state, which is R, and that has higher free energy than does either your substrate or product. And the difference in the free energy between the transition state and the substrate is called Gibbs free energy. So it is giving you a Gibbs free energy of activation, or simply we call it the activation energy. So this delta G star is giving you the value of activation energy, or Gibbs free energy of activation. So activation energy barrier immediately suggests how enzyme enhances the reaction rate without altering the delta G of a reaction. So I'll show you how it helps. And I think you have been taught this thing with the chemical reaction as well. So enzymes, they accelerate the reaction, how they attain. And so there is the presence of a catalysis is a specific binding of the transition state. So let me just come to the next slide, which shows that a diagram between the free energy and the reaction progress. So what you see is that if there is an uncatalyzed reaction, delta G star or Gibbs free energy is very high, and this is the one which is showing you. On the other hand, the substrate, when it binds, it goes to the transition state and it catalyzes the reaction, and after binding, what you see is that the free energy required is relatively much lesser. So what you need is, you need delta G, and enzyme, what it does is that it accelerates reaction by decreasing the delta G value. So see, uncatalyzed reaction required this much energy, the catalyzed reaction will require lower Gibbs free energy. And that's the way the reaction proceeds when the enzyme catalyzes any reaction. Now, how this reduces the activation energy? The energy reduction occurs because enzymes form a complex, which is enzyme substrate complex. And there is a lot of debate, and sometime I may explain this in much better fashion, is that how enzyme substrate complex formation reduces the energy. So from where energy comes? You all know that the enzyme energy cannot be generated, energy cannot be consumed. So from where this energy comes? So this is coming from any one of you? From where this energy comes, which lowers the activation energy? It is the binding energy of the substrate with the enzyme. And where does this enzyme bind? Enzyme binds at the active site. So we talked about how this binding of enzyme with the substrate reduces the activation energy. So much of the catalytic power of enzyme comes from bringing the substrate together in a favorable orientation to promote the formation of transition state in the enzyme substrate complex. So actually it is a transition state which is very important. The substrates are bound to the specific region of enzyme, and that is, I have explained, I have shown you what is the active site. Now some of you may ask how I am sure that enzyme forms a substrate complex. Why do we say so? It is said because of very important observation. So if I catalyze a reaction, I think I have a an example of that, where the enzyme gets saturated. As you have seen in case of the transport process was getting saturated with increasing concentration of your substrate or the compound which was to be transported. The same thing you find. The first clue of the here is that the constant concentration of enzyme, if I fix the concentration of enzyme, the reaction rate increases with increasing substrate concentration until it reaches to maximum velocity. And then it becomes flat. That means even if I increase the substrate concentration, enzyme velocity will not increase. So this is given over here. So if I keep on increasing the substrate concentration, initially with the lower concentration, you will find the reaction velocity increases. but after that it gets saturated. So the maximum velocity once attains. So what does it mean? It means if I physically examine this, it means the enzyme with limited concentration of enzyme, all the substrate is binding, the active site is saturated, no more. Even if I increase the concentration of the substrate, because the active site is limited, it will not increase the rate of reaction. So the enzymatic reaction, enzymatic power comes because the binding of the substrate with the enzyme. Is it clear to you?
Yes, sir.
Okay. Second clue comes that if I take enzyme alone, if I take enzyme with the substrate, you find a different characteristic of the enzyme substrate complex formation if I observe spectroscopically. So if I go to fluorimeter and I see what is the peak which is coming with enzyme, you will get something here. If I get the substrate which is serine, you get here. If I take two substrates, serine and indole, it is coming here. That means there is a difference or masking of the group when they are present along with the substrate. So that means they are changing their structure. So this gives you another example that how the enzyme substrate complex formation is involved in the reaction. Another thing which has been observed is based on X-ray crystallography, which has provided high resolution image of substrate and enzyme. So what you can do is that because substrate, if I take substrate will be converted into the product, and because the reaction is very fast, you cannot see it. So the trick has been done in case of the imaging. What has been done is that a molecule which has a very similar structure like substrate, that has been taken as an analog. So you have a substrate analog, similar structure like substrate, but it is not substrate. So substrate analog will not be converted into the product, and that is where you can see that it forms. So if I take X-ray crystallograph of just enzyme, you will find difference. If I take enzyme plus substrate, you will find a different crystal structure. So these examples shows that the enzyme catalyzed reaction involves the formation of enzyme substrate complex. And that is what shows that the enzyme substrate complex formation is essential. That means binding of the substrate with the enzyme is the primary thing which leads to the enzymatic reaction. Now coming to the point to elaborate that how the substrate and where the substrate binds. So substrate binds to the active site. This part we have discussed. And this active site is coming with the three-dimensional cleft, which is formed by the groups that come from the different parts of the amino acid sequence. And these parts, these amino acids may be far apart from in the primary structure of the protein or enzyme. What it means? It means question number one is that where the active sites are present. As I discussed in my second class, that enzymes active sites are present in a cleft. So what is a cleft? It is a kind of a burrow in the three-dimensional structure of a protein where the active site is present. Can any one of you tell me why it should be present in a cleft? Why it is not exposed on the surface? Because this cleft is the one which is creating a micro environment which is away from water. And if it is away from water, what it can do is that the reaction which is occurring between enzyme and a substrate, they will be stronger if they are by non-covalent bond. So because the enzyme substrate reaction is occurring by non-covalent bond, these bonds, they can have much better binding affinity or much better strength than if it is present in water. Why? Because if it is a hydrogen bond, or if it is your van der Waals bond, or if it is for example, van der Waals bond is initiated, whereas a hydrogen bond and electrostatic bond will be weakened by the presence of a water. So any of the bond formation is weakened, and that's why the three-dimensional structure creates a cleft, and this cleft is the one which is having some of the hydrophobic amino acid, and that's the way they provide environment which is more or less hydrophobic environment. And that's why your either electrostatic interaction or hydrogen bonding will be much better. Other than that, another concept from this figure I would like to describe is that you can see the color, for example, this color, yellow color, blue color, this color, red color, all of them, they are the amino acid. And these amino acid, though coming from different part in the enzyme in three-dimensional structure, they come close to each other and provide the binding affinity. So for example, if I open this chain and you can see here, this one, if I open this chain, you will find some amino acid which is present at 35, fifth residue, 52, 62, 101. They are the one which are contributing. That means it is not necessary that all the groups involved in the reaction should be close to each other, should be neighboring one. Now I leave a question with you that if there are only few amino acid involved, why we need so many other amino acid for the activity of enzyme? Why enzyme, nature has designed the enzyme where you have so many amino acid present to catalyze a reaction where only 4 amino acid or 5 amino acids are involved. With this question, I will end my lecture. If you have any question, you can ask. Otherwise, we will discuss in the next class. So any question? Any quick question? If there are no question, I will stop at this point and I will sign off.
Thank you,
Okay,
sir.
thank you.